Ludwig Maximilians University of Munich at Germany, and he works broadly in the area of nanobiosensors. A highly ranked researcher to his credit with several national and international funding, several scientific publications and patents, he is also a recipient of several awards that includes ICMR Extramural Research Associate Scholarships, Young Awardee from Amity University, Noida, Outstanding Plus Researcher and Faculty, then recipient of Federal Ministry of Education and Research Funds at Germany, and also many other grants from Germany. So I welcome Dr. Utkarsh Jain to this series, pre-conference webinar series, and request him to deliver the talk. Thank you, Utkarsh, for accepting our invitation and taking out some time to give this lecture to our audience here today. Utkarsh, we cannot hear you.
Sir, you are muted.
You're muted, Utkarsh.
Okay, now am I audible?
Yes, yes.
Okay. Yeah, so thank you so much, Dr. Chaitanya, for such a very nice introduction, and I really appreciate your efforts to bring this series where a lot of students who have been working on biosensor designs and developments, they will be definitely benefited for these kind of talks. I also would like to thank Dr. Pooja for inviting me together with you, of course, for this talk. So I was especially requested by Dr. Pooja that I should more be focused on the fundamentals and the basic aspects of biosensors. And so accordingly, I will deliver my talk, and hopefully I will touch the bottom where a lot of students are facing problems, and hopefully from the bottom to the advancing stage where the biosensor development is going through, and which direction we are moving further. That I am going to discuss about over the next. I am hopeful that within 1 hour time I will be able to finish. So I think that 1 hour is sufficient for me.
And then we can take question answers.
Okay, sure. Okay, so the topic which I'm going to discuss about is actually divided into 4 basic components. It is actually the direction of designing to the advancing, where each component play a very important role. So since I have included the electrochemical perspective, so of course the electrochemical perspective is dealing with where I have been working on, and since my background is with biological perspective, so I will be more focusing on biological aspects together with nano. So since nano is kind of a middle part where we are combining the biological perspective to the electrochemical convergences. So why I had selected these 4 basic components, because it actually makes a framework of whole designing to advancing. And from designing to advancing, there are a lot of things which we need to focus on. Unless until we do not know each component very effectively, we are missing somehow to really bring the kind of a idea to bring to directly transforming into the advancing stage, where our main target is to benefit the people, right? So in this sense, from designing, developing, priming, and advancing, all 4 components I'm going to discuss about. So first of all, what exactly the designing is? So you have a biosensor. Since I'm talking about specifically on the biosensor perspective, so I will be focusing on biosensor designing, where there are different components of biosensor I'm going to talk about. To develop a particular biosensor, you need to first think about what could be an analyte. So as you can see, that we are going to detect a particular component, a particular analyte in a given sample. So since if I'm working on biomedical devices, then of course a biomedical device where I'm developing a biosensing device should have a particular analyte which is a particular disease biomarker. So, for example, if I talk about a disease, neonatal sepsis, then I should be focused on what are the basic biomarkers of neonatal sepsis and whether they are significantly changing its behavior. And additionally, I need to think whether this biomarker can be utilized as an analyte in the given biosensing device development. So that is the first component I need to focus on, that what could be our target. The target as an analyte could be a protein, it can be a DNA, it can be any elemental part where we can discuss about. So that analyte, it must be focused on, if we are going to develop a device for certain diseases, then that analyte should be a biomarker. Now, the next important point which we need to think about while designing or developing a particular biosensor for a disease, then we need to think about what could be a biological component which is a binding partner or causing the effect on that partner from the analyte binding to the biocomponent. So that component is actually a recognizing component of an analyte. So suppose, as I have given you an example of neonatal sepsis, the biomarker can be a C-reactive protein. Then it's a protein, and C-reactive protein should have interacting partner as a biorecognition element, and that biorecognition element we need to figure it out. That we need to review, we need to thoroughly check whether this biorecognition element is working properly with its respective biomarker. Then we can decide which biological recognition element we can select for the development of a biosensing device. So it's kind of a first framework where we need to really work on in the beginning a lot on the designing of a biosensor, that which kind of biosensor we are trying to develop. I will discuss about this biorecognition element in next couple of minutes, but I first would like to give a brief of each component of these frameworks. Then, since, as I told you, that an analyte can bind with the biorecognition element, and this binding, this interaction should lead to a kind of an electrochemical behavior change. And that change we need to notice. So this is the basic design, from the analyte to the biorecognition element to the change what we are observing. And if that change is particularly associated or attributed to the fact that an electrochemical behavior is getting noticed, that change is our trick. That change is our idea that we need to think about, that if this change is giving certain information where we can detect and analyze that information, we need to convert into a signal to develop a biosensing device. So I guess now you understand that three major components of a design should be focused on. First, what analyte you are focusing on. Analyte can be anything. It can be, if you are working on the biomedical devices for the diseases, it can be a biomarker. If you are working on environmental perspective, it can be an element, it can be a protein, it can be, of course, a nanoparticle as well. It can be any organic or inorganic material. But the trick is, but the important point is that that analyte should bind, should have an interaction with the biorecognition element, which we need to focus on the for developing the biosensing device. And again, this binding should give certain information, and that information is important for us to develop a biosensing device. Now, once we have defined our target analyte, once we have thoroughly understand that what mechanism we would like to follow. Now, mechanism is very important in terms of when, for example, for instance, if I talk about an electrochemical biosensor, where I am utilizing a biorecognition element as an enzyme. That means that biorecognition element as an enzyme works with, or that analyte, which is actually a kind of a binding partner for the enzyme, and a product is formed. It may be that the product is not useful for us, but during this reaction, some electrochemical change what we observe maybe in the form of electron generation, that electrochemical change that we need to focus on for a particular sensing mechanism. So depending upon the selection of a biorecognition element, we can decide the mechanism which one we are going to select in our design, right? And accordingly, the next development should be working on. Now, since the selection is very important, and based on the selection, we can work on the mechanism, and finally the analyte can be detected and give the information on the electrochemical signals. And these electrochemical signals can be converted into a detectable information, where we can transform this information in a form of a device. The next part is, talking is very easy, that okay, we have developed a, we have designed a biosensor, but the most tricky, most difficult, most challenging part is the development. So in each stage during the biosensor development, we need to focus on many parameters. For example, if my analyte is, say, a protein biomarker for a disease, for example, X disease is there, and I am going to detect that X disease through a particular biomarker Y, which is present in the blood. In that case, Y is my target analyte. Now, I need to figure it out that the protein should have binding or should have interaction with my biorecognition element which I have selected. And in developing this stage where biorecognition element which I have selected, once it is already bound to the analyte, then I need a particular approach where, since I am working on the electrochemical sensor, then we need a particular electrode where I have to code this biorecognition element, and because of this, electrode binding to the biorecognition element can give the signal on the electrode which I am going to measure about. Now the thing is, when you develop, when you fabricate these layers of selection, then in each stage you need to characterize. Number one, if you want to coat a particular protein which binds the biomarker, then that protein should be properly and uniformly distributed on the electrode surface, specifically on the working electrode surface, to get a detectable signal. And this uniformity, this approach should be properly characterized in each stage while we are developing a biosensor. So, of course, the immobilization procedure and other characterization stages where we need to really focus on comprise the development part. In some cases, in actually most of the cases, what we have been working on, we are working on the nanomaterial based biosensors, where we specifically select certain nanomaterials, advanced and functional nanomaterials, where we make a middle layer from the electrode to the biorecognition element. That middle layer is of nanomaterials. The reason I'm going to talk about in the next couple of slides why we selected the nanomaterials. Just in a nutshell, I can tell you that nanomaterial can give you a proper more than the threshold value, so that we can get detectable range. For example, if I'm working on a current-based detection, and my electrode material is not giving that information which is detectable, then in that sense, I need to coat certain nanomaterials after the selection of those nanomaterials. These nanomaterials can enhance the conductance on the electrode surface and give the more information and detectable range where we can really detect a particular signal. This is actually very important. Why? Because, say, if you have a biomarker which you want to detect, and that biomarker is kind of a trace element or very low concentration presence in the sample. Now, the thing is, since the presence of that biomarker is very low, and on the electrode surface when it bounds with the biorecognition element and giving a particular signal, and that signal is not detectable since the concentration is very low, then that signal cannot be converted into a proper, say, where we can tell that this is the particular concentration of that particular biomarker. In that sense, nanomaterials are really useful, that they amplify those signals, multiply those signals so that we can get the information properly and we can convert that signal into the detectable form. So a couple of parameters we need to think about while developing a biosensor, and each characterization steps are very much important to get an optimized sensing performance of a biosensor. So we have discussed about design, we have discussed about development, and this is the one which we have more focused on and which we have actually developed this idea of priming. And this priming is not at all used before in biosensing devices, and very recently I have incorporated, because I have noticed over the years that the students or the people or the researchers who try to develop a sensing device related with electrochemical sensing, they miss this part, priming. And because of this priming approach, you actually cannot convert your developed device in a really advancing form, or since there is a gap where you cannot just develop a commercial device. Priming is a bridge which fills the gap from your lab device to a commercial real biosensing or medical device. So you can see this is very, I don't know, I should say that this is very sad to hear that most of the people who have been working on biosensing devices, they make the device, but they are really not converting that device into a final commercial product, or they don't translate that part into a real commercial device. And I can tell only 0.5% the real biosensing approaches are convertible at this moment, converge into the biosensing devices. So we need to focus on this priming part a lot while developing or once we have developed a device to convert into the commercial perspective. Priming is actually a word which is widely used in biology as well. So say if you know that polymerase chain reaction, PCR machine, we use the primers. We have priming word in psychology as well. We have priming word in couple of different disciplines as well. But here the priming is actually the start or initiation of a particular convergence, where once you have started that part into a, say in our case, into an advancing or commercial stage, that initiation we require. So it requires a lot of optimization. Optimization is one part of priming. And we need to focus on couple of parameters, including the, say, limit of detection, sensitivity, response time, and couple of things in a real form where the lab form is converting into a what we say, a form where we can really use them properly. So I'm going to focus more on priming in the next couple of slide as well. But first, you need to know that optimization, troubleshooting, they are all the part of priming. And advancing, of course, this is not new, but this is a very dynamic process that even if you have worked 5 months before on one project where you have developed a device, unless and until you do not continuously make a proper advancement in the device, it will be old dated. It will not work at all. So in most of the cases, proper validation, proper incorporation of those parameters, those materials, those ideas in your biosensing device will make a proper advancement. So these 4 framework, if you have a mindset of working accordingly, this I think will definitely get benefited to you, and this will definitely make a proper system of making a biosensing device, incorporating all designing to the advancing parameters. So let's talk about a bit more about the designing. I have discussed about the analyte, that analyte can be an inorganic materials, it can be an organic materials and inorganic materials. Organic materials can be, say, if I'm going to detect a bacterial infection, and that bacterial infection, say, Mycobacterium tuberculosis. So Mycobacterium tuberculosis is a bacteria which causes the infection and causes the tuberculosis in lungs. This bacteria releases mycotoxin in the lungs, and I want to detect mycotoxin in a biosensing device because I don't want to go to the laboratory and wait for, say, 2 days. I should have a proper device where even a normal person or clinician can take the sample and load it on the electrode surface and can provide the information that this bacteria is present in a sample. So this is the case where certain toxins which are released by the bacteria can be detected. As I told you, that it is not restricted to just one perspective where you are dividing your biosensing devices into the biomedical or biomedical or healthcare perspective, but environmental perspective where the you detect the pesticides concentration, and another part where a lot of environmental composition can be detected. Antibiotics can also be detected. So all the organic materials, if I'm using a terminology, if those organic materials have an interacting partner and the improper interaction with a biorecognition element, then only I can utilize that part as a development for or designing for biosensing device. You can also detect certain metal, phosphates, and nitrates. So actually, that is not my area where I'm more working on the organic materials. Now, the biorecognition elements, what it can be, that's very important. So again, biorecognition elements are very specific. It can be an enzyme, as I told you in just my previous slide, where enzyme binds with an analyte to give the information, and during this process from enzyme to the substrate binding, when the product is formed, certain electrons are generated, and then those electrons are over the electrochemical signals, and they are actually proportional that how much substrate concentration or analyte concentration are present in the sample. It can be an antibody. So I'm not sure how many of you are from biology background, but I'm understanding. I have an understanding that most of you are from biology, from physics, or chemistry, because it's an interdisciplinary branch. So an antibody is a kind of it's of course immunoglobulins, proteins, which have a very high specificity to certain antigens. And in the case of antibodies, we can use antibodies as a biorecognition element, which can bind with certain analytes as a form of antigen. And this binding can lead to give the information on the electrode surface that binding can give the or cause the impedance on the electrodes to give the final information where we can get the signal that that particular molecule as an antigen is present in a sample. It can be a nucleic acid. It can be a whole cell as well. So this is actually new idea that not lot of people are working on the whole cell biosensing devices, because this is actually a very difficult task to do. But on the other hand, it can give a proper physiological information where the whole cell is giving the information, where the change of behavior is observed on the electrode surface. It can be, the biorecognition element can be an aptamer. So I have worked with the aptamer as well, and I will show you 1 or 2 slides of the aptamer, how they were developed. So aptamer is actually, let me tell you, the aptamer is actually a kind of a spontaneous sequences. It can be a sequence of 50 base pairs of DNA. It can be of 50 base pairs of RNA or protein that have a normal tendency or affinity to bind with certain molecule. So for example, I have a peptide sequence or protein sequence of say 20 kilodalton, and this 20 kilodalton have a particular region where if I mix particular sequences in the sample, and if they are naturally or spontaneously bind with those area, those protein conformations, then these sequences are called the aptamer sequences, and they are very selective. They are very selective to that particular specific molecules, and that is our task, that I need to figure it out which biorecognition element I should select. And aptamers, since they have a high selectivity towards certain molecules, it can use as a biorecognition element. And a lot of people have been working, and we were so lucky to have the collaboration from NASA people who were working on the aptamer technologies for detecting certain psychoactive drugs which we had worked on. Then certain peptide sequences, they are actually the binding peptides with another molecules where they show the tendency of effective binding. And this effective binding I should indicate in the form of a selective and specific binding. So non-selective binding cannot give the real biosensing device, because the selective or specific binding is very important in terms of developing the biosensors. Certain bacteriophages, because these are viral particles, and they have a very strong tendency to select the bacteria. So you know that the bacteria are having specific bacteriophages, and these bacteriophages are having the certain proteins on their tail. And when they bind with the bacteria, since they have a very specific binding, so we are trying to utilize, this is our new work which we have been working on, to detect the bacteria as well as certain bacteriophages to use as a bacteria for the biorecognition element. So we are still working on the bacteriophages as a biorecognition element. And one of the work which we have been doing is on polymers. So the polymers are actually these molecularly imprinted polymers. So I will show you some of the slides as an example that how we developed the biosensors or biomedical devices through utilizing a particular or specific molecularly imprinted polymers in our devices. Signal, once we want to transduce, in electrochemical sensing, we use the electrochemical signals, but in certain cases, if you are working on optical sensors, then you use the opticals and magnetic and other parameters. But I'm going to only focus on the electrochemical sensors. So this is again, I have already discussed this part that you should have certain analytes, then you should have the biorecognition elements. Then come to the part of nanomaterials. So actually, the problem is when we have an analyte, and of course we have a biorecognition element, this binding from the analyte to the biorecognition element, unless and until it is not giving a signal, it's of no use for us, right? So in that case, we need to select a proper transducing system where the electrode, say electrode can be a screen printed electrode or disc electrode, that I'm going to discuss about it as well. But once the electrode is there and the binding of biorecognition element on the electrode is occurring, and if it is giving an electrochemical response, but the response is not effective, then I need to use these nanomaterials to enhance the signal. And that signal is actually should be detectable signal. So a couple of advanced nanomaterials have been used by a lot of research groups, and they are used in glucose detections. Since I have utilized the glucose detection platforms as well to develop the HbA1c glucose, as well as certain albumin present in the blood. So I will show you that part as well, where we have developed a small biosensing device which can be connected to the mobile app, and through the mobile you can have a proper detection of diabetes. They are actually advanced nanomaterials. They are actually used in imaging and couple of more drug delivery, therapy, cancer detection. But in our specific case, on the signaling part, where we want to amplify the signal through the nanomaterial coating, that I'm going to focus on. Now the thing is, why nanomaterials? Why not the bulk materials? What property nanomaterials they have that is not noticeable in the bulk materials, and why only nanomaterials? So to give you an answer, that nanomaterials are very effective when I want to see a change electrochemical signal, enhance the electrochemical signal, amplify the electrochemical signal. And apart from enhancing the electrochemical signal, since they have a very high surface to volume ratio, and if my biorecognition element is very small, in for instance, if it is an enzyme, that since the surface area is very high, then the loading or binding, or I would say coating of enzyme on the electrode layer is a bit higher, and that loading is uniform when I'm using the nanomaterials. Secondly, if I'm using the couple of nanomaterials which have a bit different properties, but they are widely used as a composite, then they may show the synergistic effect that they multiply their amplification signal while detecting the information. They are easy to utilize, since this procedure is not that much difficult that you need to load or immobilize or coat the nanomaterials on the electrode surface. And they really are having a kind of an approach where the sensitivity is strongly noticed. Okay, so that was the part of biosensing. And as I told you, that electrode which we select in the form of certain screen printed electrode. And in the traditional way, when we talk about a proper electrode setup, then I can show you that this is an electrode setup which we used to use previously when the screen printed electrode were not being utilized as they are utilized in the present day. So they have actually 3 type of electrode system. So we actually work on 3 type of. There are 2 types of electrode, 4 types of electrode system as well. But we usually work on the 3 electrode system, where the working electrode, reference electrode, and counter electrode are embedded in the system. And the coating of biorecognition elements and the nanomaterials we actually do on the working electrode, because this is the one where we need to focus on, where we have all the coating and possibility to get the signal. Reference and counter electrode, they complete the circuit and have the proper circuit in a way that we get the electrochemical change which is noticeable in the form of a signal. So this is kind of a electrode setup, and this is a very small electrode sensing potential state device where we connect the screen printed electrode, and then we load the sample right here to get the signal. Now, part which I should be focused on, that the development part, right? The electrode, when it is coated with the nanomaterials, we need to have a proper selection, whether we need to develop an electrode surface with certain nanomaterials, say, if I want to work on the graphene, I want to work on the 3D nanomaterials as graphite, carbon nanotubes, and couple of more carbon associated nanomaterials. So this is just one example. There are enormous functional and advanced nanomaterials based on their specific properties we are going to utilize. For example, nowadays we are using MOS2 and Maxine because they have a 2D, Maxine have a 2D layered materials where we can effectively coat a particular biorecognition element as compared to the other nanomaterials, say graphene. So they have shown. So it's a hit and run trial where you need to really work on certain nanomaterials with your electrode to check whether the electrochemical response is that effective or not. And once you optimize that sequence, then only you should go for the development. And during this process, you need to focus on certain characterization procedures where you need to verify that the electrode have the proper and uniform and up to a certain defined limit and the layer of nanomaterials coating. So each step is required to develop a biosensor when we have been working on to finalize the development and the fabrication of a biosensing device. As I told you that the working electrode is the one which is our main target. So a couple of screen printed electrode, microfluidic devices and disc electrodes are being utilized for coating of nanomaterials and then coating of biorecognition elements, and then finally working on to optimizing and priming the sensing devices. This part is not that much important, but we need a potential state where we need to electrodeposit certain nanomaterials and couple of procedures which we follow. If you have been working on a, if you are working on an enzyme-based biosensor, then you need to coat on the working electrode these enzymes, and sometimes you need to have the covalent linking, which is more effective. But to verify, to get the preliminary signals, you can check or screen through the drop casting, where you load the proper drop arrangement on the working electrode of an enzyme, and then you can get the sample. Although I would not recommend the drop casting because it's not that what we say scientific in terms of whether you really get a proper or the signal which you should get. So covalent linking is much more effective than the drop casting. But for the preliminary study, preliminary data information to check whether the electrochemical behavior response you are getting or not, then you can go ahead with the drop casting. Priming the part, as I told you, that it's the initiation of the optimizing parameters and couple of things you need to work on while you have. So finally, you have electrode surface, you have decided the nanomaterials, you have coated the nanomaterials, and then you have coated the biorecognition elements, and then you are detecting a particular analyte in that. While having these parameters, you need to have certain factors which should give all the information about that biosensing device. Those parameters can be what signal you are getting, what concentration it is able to detect, what is the minimum concentration, what is the minimum signal it is going to detect, For instance, if you are working on a glucose sensing device, and since glucose is abundance in our blood, and the enzyme-based sensor glucose oxidase, when it is with our device, which is coated on the screen-printed electrode, then the enzyme concentration should be properly measured, and that enzyme concentration, while having a particular area on the working electrode, how much glucose molecules it is binding, and what is that limit of detection, whether it is a sensitive approach or not, and what could be the sensitivity of that particular reaction, what is the response time, that I don't want to wait for 3 to 15 minutes. Maybe in 3 to 15 minutes, I'm not going to get the right signal. But in enzyme-based biosensor, I can get in the, say, 30 seconds. So what is the response time? I need to check. Response time can also correlated with the fact that your response is differed in each timing. So you need to optimize that parameter by priming all the electrochemical device development scenario, where you need to focus on the stability of your performance. You need to focus on the whether you can recover the sample outcome when the information is really accurate. This is one of the most important parameter, where you need to check whether the device which you have developed is actually giving the same information or closer information to the gold standard method. That means if some analytes are detecting through the HPLC, so HPLC as a gold standard method, I need to focus on that my data, my information, electrochemical response is actually correlating with the HPLC methods or not, right? So a couple of more things I need to incorporate in the priming while developing and while transforming the developed biosensing device into a real device. Now the part is advancing. I'm not focusing on that part because it's of course a futuristic approach, but just I can tell you that nowadays automation and through AI-based sensing or AI-based data processing can lead us for more advancing biosensing approaches. One of the major and the basic concept of advancing comes from the real working electrode. So if I have a working electrode and I want to make an advancement on the working electrode, one should focus on that part to bringing advancement in future. But the working electrode, when say I was working on the one nanomaterial, say carbon-based nanomaterials, and I have noticed that when I have a MXene as a layered material, then the amplification is much beneficial in terms of when the layered MXene I'm using on the working electrode. So in that case, you are advancing the working electrode responses and nanomaterial responses to the more advanced materials. So this is just one example where you need to constantly advancing your approaches to eliminate the previous one in terms of if you are facing any challenge to a particular priming parameter, and that priming parameter you need to determine. And once you have determined those priming parameters, you need to constantly working on advancing approaches. And the future is, of course, one of the futuristic approach is wearable sensing devices. I'm skipping this part again, but I need to tell you that nowadays the wearable sensing devices are much more transforming as compared to the non-wearable sensing devices. Now come to the part of what we have been working on for a couple of biosensors over the couple of years we have developed. So to start with one of the example, I'm sure I'm not getting too late, but I go very fastly. So to give you a very basic understanding that how a biosensing design can be done through a proper physiological approach in our body. So as you can see that this is a acetylcholine part, where acetylcholine is the one which is a neurotransmitter. So we were working on one neurotransmitter, acetylcholine, and we wanted to develop a device. If somebody is having mental disorders and some couple of acetylcholine related disorders, including the Alzheimer's and Parkinson's, then acetylcholine information is very important. Since we wanted to develop a device of acetylcholine, we wanted to have a physiological parameter in real sense, where you have a kind of a nerve nerve to the muscle junction, and you can see that suppose this is the muscle and this is the nerve, and acetylcholine is actually be resided in the between the junction, and when the acetylcholine concentration is higher in the junction, it binds with the receptor which is located on the muscles and gives the proper response. So as you can see, the calcium is actually very important once it is internalized inside in the nerve. Some synaptic vesicles are formed and acetylcholine is released in the junction. In the junction, there is one enzyme, acetylcholine esterase, which is present in this junction, which binds with this substrate as a acetylcholine, and once it binds, it releases the choline, and this choline is get back to the nerves, and this is kind of a recycling mechanism where acetylcholine is constantly reutilized. So in this case, since we wanted to develop a device for the acetylcholine, we utilize the acetylcholine esterase enzyme as well as choline oxidase enzyme as co-immobilization to develop a device which detects the acetylcholine. So as you can see now, this is the graph. Most of the electrochemical PhD students, they know this graph, that this is the cyclic voltammetry, this is the impedance, electrochemical impedance spectroscopy. So in the cyclic voltammetry, as you can see, that as I told you, that we need to have a proper proper identification of proper characterization of all the stages, from the electrode to the coating of nanomaterials to the coating of enzymes to the detecting with the substrate, and in enzyme-based biosensor what signal we are getting. So as you can see that this is the oxidation peak of a cyclic voltammetry, and this is the reduction peak of a cyclic voltammetry graph. Now, if you see this graph, the black one, what you see is the bare electrode, bare gold electrode, right? So bare gold electrode is giving certain current, which is in the form of a milliampere on the electrode surface. Now I start coating of nanomaterials to the biorecognition element. So here there was a composite with the gold nanomaterials with reduced graphene oxide, and once I have coated, electrochemically deposited the reduced graphene oxide on the electrode surface, you see an enhancement of a certain current, which is indicated in the red graph, right? Red line. Now, there was a composite MnO2 multi-volt carbon nanotubes and reduced graphene oxide, and when they were all present on the electrode surface uniformly, then the blue one, as you see, that they have significantly enhanced the signal. And that signal, as you notice, that nanomaterials can really amplify the signal in the detectable range. Now you coat the acetylcholine esterase and choline oxidase. These are two enzymes which are co-immobilized on the electrode surface uniformly. So once you coat, then you see the signal that the pink one is really dropped significantly, because they are not allowing us to passing the current, and they are causing the hindrance, or the current is not getting detected because of a biological molecule. Now, what is the most important thing? That when we add the acetylcholine in the sample, since acetylcholine esterase and choline oxidase as an enzymes are present, they immediately get reacted, and in this process the electrons are generated. And you see from the orange line to the green line, you see a very amplified signal, which is up to a certain limit, having some limitations, which is proportional to the number of acetylcholine molecules present in a sample. And you can actually have the different concentrations evaluation through this cyclic voltammetry approaches. And you can also verify through the electrochemical impedance spectroscopy, EIS, which is opposite to the cyclic voltammetry, where you get the lowest graph when you have acetylcholine esterase to the sample which is present. Now, that was the part of one enzyme-based sensor. Now, one of the sensor which we developed is a polymer-based sensor, where we developed a molecularly imprinted polymer and electrode deposited on the electrode surface. When the molecules are present, when we load the molecules on the polymer surface, then these molecules are getting entrapped on the polymer surface, and then you check the electrochemical response. And whether the molecules of that specific molecular imprinted polymer surface are causing any resistance or impedance that is detectable in the electrochemical signal. So that we are going to notice. So how to develop this kind of device while we need to prepare a molecularly imprinted polymer? So what we do, we prepare the molecularly imprinted polymer by adding certain composition. It's not very easy, it's very difficult process, because we need to optimize the well-defined composition of template to the monomers to the polymerization initiators. So what you do, you take the monomers and you mix the monomers with the template together with the cross-linker polymerization initiators, and you polymerize the template molecule and the polymerize the polymer. Once it is polymerized, you remove the template. Once you remove the template, since the template is of a molecular size, that template, once it is removed, it actually leaves the imprints on the polymer surface of same molecular conformation of that particular molecule. For example, it's very simple, the sieves which are formed. So if I wear a shoes, right? So my shoes is for my number. And if I develop my shoes, then if I remove my leg out of my shoes, then the size of that imprint of my leg is remained there, right? So that imprint, that sieve, when we remove the template out, it is actually present on the surface or the polymer. So you see the molecular size patterns are appeared on the electrode surface, and once we electrode deposit, and when these uniformly layered polymer surface are on the working electrode, and when the molecule are again reloaded, or when it is present in a sample, it will go and bind there. We need to characterize that whether the imprint what we have formed are actually the imprint of the molecule. Then we perform the BET analysis to check the surface area of that particular sieve or imprint which is formed. So we had worked on a couple of molecules, neurotransmitters and certain other biomarkers, and we really worked very well in terms of getting the signals. And this is another example where we worked on serotonin and dopamine for molecularly imprinted polymer detection. So how it works once you have developed a sensing platform utilizing the molecularly imprinted polymer. So for example, this is acetylcholine template when we assembled and properly developed a molecule. So as you can see, this is the electrode surface. This is the polymer, molecularly imprinted polymer which is developed. And when, so it is kind of a biorecognition element. As I told you that A polymer can also be a biorecognition element if it has a tendency to selectively bind to a particular molecule. So it's kind of a open and closed conformation. So whole layer of polymer on the electrode surface now is having that particular molecularly imprinted polymer without any template. So they are all being associated in a single layer having the sieves of a same molecular size. Now suppose I have a sample, and in that sample I am going to detect a particular molecule for which I have developed a molecularly imprinted polymer. Then this sample having the molecule will go, and these molecules get entrapped in the sieves which are specifically for that size, and they close the whole conformation after the washing. So once it is closed, all the molecule get entrapped because they had the same imprint, and they cause the impedance on the electrode surface which we detect before and after adding the molecule, and that can give us the information. So as you can see, in this process we can have actually the possibility of developing a particular small electrochemical strip-based device where we have on the working electrode a polymer specifically designed for that particular single molecule for which I am going to develop a device. And that signal can give us a thorough information within 2 to 3 minutes for the entrapment to the washing. And as you can see, that it will eliminate most of the processes where you don't need to perform the HPLC and lengthy biochemical assays. And with this entrapment of a molecular size on the polymer can give the thorough information for the transformation. One of the most interesting story when we were working on certain neurotransmitter, we wanted to develop a device for anandamide. So actually, we were funded by SERB, and this was one of the most ranked project, and SERB really gave good ranking for this project, excellent ranking for this project. The idea was that anandamide is actually a neurotransmitter, and you know, Anand is a Sanskrit word. It means happiness. And it is the most recently discovered neurotransmitter. And the people who were thinking about anandamide as a neurotransmitter, they actually developed this device because they wanted to check whether if somebody is having or taken marijuana, then why marijuana makes people so much happy that they are in a different world, right? So that was actually funny that when a person who has taken marijuana and he laughs one time, then after certain minutes or over the period, he continuously laughing on certain aspects. So this is actually a reason that the structural similarity of THC or marijuana is having with the anandamide. And this is intrinsic neurotransmitter which is responsible for our happiness. Anandamide concentration is also, as although it's a neurotransmitter, but this molecular concentration is also present in the chocolate. When somebody is eating chocolate, they feel very happy because they enhance the anandamide concentration in the body, and that will make you happy. So we also developed a device with the for the anandamide detection. And anandamide is not only restricted to the happiness, but it is also being utilized in the case of a lot of disorders as well. And now people are still working on to identify what is the reason that anandamide have so much of different physiological functions. So this is kind of a bulk. So we worked on the molecularly imprinted polymer device for the anandamide, as I have told you, with other neurotransmitters. So this is kind of a bulk polymerization. I'm not going to discuss in detail. It is just the kind of a representation where you need to add the template molecule, EGDMA, and then EGDMA as a cross-linker, and the monomer, and then how you prepare a polymer. After the polymerization, you need to check whether the imprints are really formed or not. As you can see that the A, C, E, and F. A is actually the anandamide with imprinted polymer. And when we remove the anandamide while developing the polymer surface, then you see the imprint of anandamide molecules are beautifully arranged. And that is actually the information that when an anandamide is present, they will go and entrap on the polymer surface. Another work what we have done with the electrode surface is enzyme-based biosensor for HbA1c and glycated albumin, and both were with the chronoamperometry. Chronoamperometry is actually a process where electrochemical process where we use the current as a measurement of a particular sensing device. So this is the device which we have prepared, a very small device when it is connected with the on one side with the electrode and on the other side with the mobile. Then all the information you can get in the mobile for checking the HbA1c and GA. So how this device was developed, it is actually the electrochemical enzyme-based biosensing device where the working electrode were coated with the different enzymes. And of course, certain nanomaterials were also utilized in this process on the two different electrodes. So working electrode can also be not a single working electrode, but it can be a channel of working electrodes, say 2 to 4 to 5, and couple of working electrode you can utilize for the different molecules. So if you are working on the multiplex biorecognition, if you are working on the multiplex devices where you have different biorecognition elements coated on the different working electrodes, and you can detect through certain approaches, you can actually detect 4 to 5 couple of biomarkers for a single disease. That is actually the future advancement what people are working on. So when we perform the nanomaterial coating, then we, as I told you, that each step we need to characterize through certain approaches. SAM is one of them, that we need to verify whether the coating has been done of the nanomaterials or the biomolecules or not. So you can see that SAM images where certain nanomaterial, this is the bare nanomaterials are coated, and this is the layer of an enzyme which is coated on the electrode surface. So this actually give a preliminary information that the layer of enzyme is coated on the electrode surface to develop an enzyme-based biosensor. So this was the device which we developed, and this is really now we are transforming, trying to make it commercial, and certain validation are left. And this was actually funded by BIRAC, and BIRAC really supported, and they are giving us for next grant for validating this device with the clinical hospitals. Another work which we had developed for that was the psychoactive drugs. So this actually worked with the APTA sensors, aptamer technologies, with the NASA professor Donald S. Burke, and he really liked our idea because he was the expert of actually he is the expert of aptamer sensing technology, aptamer technology. And we were the expert of, not expert, I would not say I'm an expert, but electrochemical sensing approaches. So he wanted to combine this approach where he wanted to check whether the aptamer can be utilized in a sensing devices or not. And it came to our idea that there are a lot of extraterrestrial biorecognition elements, for example, aptamer, which are present extraterrestrially, and he could brought these extraterrestrially biorecognition elements, for example, in the case of aptamer, to check whether the binding of these aptamers are occurring with certain molecules or not. And luckily, we got that certain molecules, for example, the amphetamine, methamphetamine, and MDMA. These are the psychoactive drugs widely used in the US. and they are clinically prescribed drugs, and certain medical uses they have. So couple of commercial perspective of amphetamines are widely used. So he wanted to check whether we can have these aptamers loaded on the electrode surface, and can we develop a sensing device for this or not. So as I told you, I have already described this. What is aptamer? So there are certain sequences spontaneously binding with the particular molecules, and this is actually the spontaneous. So for example, if a molecule is placed in n number of base pairs, and once these base pairs are rearranged in a fashion that certain base pairs will show a very specific sequence selection for binding to the specific region of the molecules. So these are actually the aptamers. So these are very, very advantageous because they have very high affinity, excellent affinity, and they have a strong stability. Although the synthesis of aptamers are not very easy, it takes a lot of processes. One process is aptamer Salex process. One process is what we call aptamer synthesis process, is Salex process, and it takes 2 to 3 years to synthesize even a 50 base pairs of sequences for a particular molecule. So now you can understand that how much tedious job is it for the aptamer sequencing, aptamer synthesis, and then finally conclude that it is having high affinity to a particular region. So what we developed two types of sensor: amphetamine apta sensor, and another one is MDMA apta sensor. And we used the gold nanoflux, and then we coated the gold nanoflux with the aptamer sequences on the electrode surface, and we checked through the electrochemical sensing whether the amphetamine is effective, since we knew that the aptamer was specifically for amphetamine. So amphetamine standard samples were properly evaluated, and they were giving the really great information. We published this in the paper Surface and Interfaces. And another one is the MDMA, for which we utilized the tin nanoparticles and then coated these tin nanoparticles with the aptamer sequences, and then we finally developed an apta sensor for the MDMA. Last but not the least, I also would like to tell that biosensing is not restricted to the biomedical devices, but bio-related sensing. We are developing a library where certain electrons can also generate electricity, or certain bacteria can also generate certain electric current, electricity. And we are making a library in collaboration with one professor from US, and we are making a library for bacteria who are having certain responses to the electrochemical sensing. So one of the professor from University of Massachusetts, and Dr. Derek, he is working with us on this microbial fuel cell approaches, where we are preparing a library for the electrochemical microbial fuel cells, where the bacteria can be utilized as an electrochemical in the electrochemical cell, where the electrons are generated by the bacteria. So how we do? We collect a lot of samples around the Uttarakhand region, then we isolate these microbial species, we preserve them, and then we check one by one what and how much currents they are producing in a particular concentration of certain elements. And then once we have identified a couple of species which are generating, one species is Geobacter, which is generating the electrons, and we are using those species which are referred as the electrogens. We have named these electrogens, the new name, and these electrogens are utilized on the electrode surface to prepare a microbial fuel cell, and they are actually giving very good responses. So certain modification is Derek doing, where he is preparing certain genes where the electric current generating genes species, he is trying to prepare the what we call vectors, and then we are preparing certain strands which are having the possibility to generate a higher electric current. So that then we will utilize those bacteria in our processes. So one of the finally one of the case I would like to think about that since if I suppose giving you a disease, say neonatal sepsis, so how you will transform the disease parameter to develop a biosensing device. So I would like to let you think about it in this perspective, from the designing to the development to the priming and advancing. What exactly the pattern should be, what exactly the approaches and the direction should be from the neonatal sepsis development to the device development, where you need to figure it out certain biomarkers, whether the biomarkers are really feasible to develop a device. And finally, when you come to the development stage, how you utilize the priming stage, and finally the advancing stage to make it in the future ready solutions. So this is the one which I leave you, which I let you think in. Maybe you can contact me in future, how you think, or you just give do or case study or exercise you can do with with this particular disease. And then once you are well versed with the idea of 4 framework, from development to the designing to the advancing, then I can tell you that you can really develop a required, which is truly required kind of a biosensing device, medical device, or any other sort of sensing device in your work. And you need to be really sure what parameters, what characterization steps you need to follow. Okay. So I have taken a biosensor technology initiative in our university, and since this initiative will help people to learn about development of biosensing devices, maybe there could be certain idea, but if there is no framework of proper development and advancing, then of course you cannot work on. And ultimately, our focus is not just to publish a paper, not just to develop a device, but really to make it as a market ready solution, and to bring to the people where we can really reduce a lot of load of hospitals to the devices which we can utilize as the home-based devices. So there are a lot of possibility which we can use as a wearable and home-based sensing devices. Finally, as I told you, that wearable sensing is the future one where a lot of people are working, and we are also into this direction now, that how we can transform this screen printed electrode to the real wearable device. So recently we developed a device, uSense, and we are developing certain approaches where we are transforming this uSense device to the wearable sensing device. So that is our future focus where we can be working on. So this is what I actually would like to pointed out, that I am not a biosensor specialist. My background was, when I was doing my PhD in 2008, my background was cell biology, microbiology, and immunology. And I was quite fascinated when I joined Indian Institute of Science, Bangalore. And I noticed that a lot of people who don't have the biosensor development idea or biosensor research perspective, they are lacking actually the real biological theme. So I thought that why not to transform my fundamental idea, fundamental research exposure to the applied research where we can reach to the people. So that was the idea where we previously thought in 2012, 2013, I thought to transform myself in the applied research, and then we moved further. Although my heart is for the fundamental research related to the biology, but I changed my time to time I learn fundamental things, and then my mindset is then how I can transform my fundamental knowledge to the applied research. So I was, since I was in LMO Munich, and my professor was Dr. Rainer Haas, and I worked there till 2012. Then I joined a couple of couple of places. I worked in Zeiss, Carl Zeiss. I worked in cell culture laboratory in one big biological company as a group leader, and then I got bored there because I wanted to do certain different things academically to give my ideas, and then I joined MIT University as an assistant professor. I remained there for 7 years, and then for 3 years I moved to UPS, and I have my own research lab right here. Luckily, we were so fortunate that Government of India, with approximately 9 funding agencies, we got the funds, and till now 5.4 crore grants we have got, and this is the support from couple of we got the UKRI grant, we got the BIREC grant, SCRB, DST, DBT. Except the ICMR, we have most of the funding agencies who supported it. Actually, ICMR I did not apply it so much. Nowadays, ICMR are supporting a lot of on the biomedical devices, and luckily we had amazing collaborators. We had the collaboration from the Nobel laureate Barry J. Marshall, who gave the idea of developing a device for Helicobacter pylori. And we have the collaboration with Malmo University, Sweden, Pasteur Institute, Paris, and of course IISc Bangalore, and one of the NASA professor, Donald S. Goodkin. I did not include most of our collaborators. I just show the work which we had done from 2016 to 2022. So this one work was actually done in the MIT University, and we had more new responses, new work coming up in line. And fortunately, the Donald, he is also at the moment associated and want to work again with us. The idea I want to focus all of you to think about one perspective, that don't always think that whatever you had started, it will go as it is. In the research, it's not like this. You need to constantly update in terms of advancing. And previously, maybe you have thought to make a circle and it has become, a square. It's not always happened. So you need to figure it out, the solutions, where can you convert this square into the round shape? And it's a constant process. So I would recommend to my fellow PhD student and beautiful, hardworking PhD student that don't get depressed when you do not get result. Maybe you need to think some other perspective, some other mindset where that particular experiment can work in different directions. So that will definitely, I can tell you, definitely give you certain information that where you can move further. And moving is gaining. Once you move forward yourself in a proper mindset, you can, of course, gain a lot. Thank you for your attention. One more thing I would like to tell in the last, I'm sorry I'm getting so late, but we recently got the Sparsh BIREC grant to open a Sparsh Center. And a Sparsh Center is funded by BIREC. And we got the 4.2 crore grant, in which we will be working on 3 main domains. One is climate resilience, farm to plate, and public health. And we will be hiring 5 fellows in each cycle. So total number of 15 fellows we are going to hire, and who will be working on bridging the gap between the laboratory or the problems which are actually the social problems related with the public health, farm to plate, and climate resilience. And we will immerse them, we will guide them, and we will develop the products, and finally we will try to bring and implement those in the market. So that is our target, that during the next 5 years we will be working on 3 domains to really bring a change, bridge a gap to fill where we are not transforming the laboratory models to the commercialization. So I'm hopeful that if you want to work on these perspective, if you want to work in certain related with these ideas, climate resilience, farm to plate, and public health, we will be really happy to incubate you in our center. Okay, so thank you so much. I am hopeful that you understood a lot, and I'm now open for questions if you have, and I will try my best to give you answers.
Thank you very much, Dr. Utkarsh, for that informative and engaging lecture. Starting from the basics and then biosensor application with their mechanism, and of course, your interesting research on anandamide. I would like to say that your journey is very motivating for us. I'm sure the students and participants have greatly benefited from your talk, and they must have many questions in their minds. So I will ask the students to write their questions in the chat box, and I can read from there. Any questions you can write in the chat box. Okay, in the meantime, I personally have a question. Sir, you talked about the magazines that you used magazines for in the sensing application, and you also discussed about the mechanism with enzymes and polymers, how they sense the analyte. So, what is the mechanism with magazines?
So Maxine is actually again a nanomaterial which amplifies the signal. Uh, for polymer-based and enzyme-based sensing, when we do not use any nanomaterial, we are not getting that much detectable signal. So previously, when we were working on graphene sheets or certain other nanomaterials, in the case of comparatively the Maxine, the layered Maxine can give a significant enhancement in the current as compared to the other nanomaterials. And that is the recently which we have evaluated, we have screened the Maxine and MOS2 to get the electrochemical signal in higher quantity, and that was actually significant. So that's the reason now we are thinking about Maxine to transform in the electrochemical devices. Another reason that Maxine is much more efficient when we want to load or immobilize biorecognition elements such as enzymes on the Maxine surface as compared to the other nanomaterials.
Okay, so basically, magazine enhances the signal.
Yes, exactly. Amplify the signal in a greater terms as compared to other nanomaterials.
Yes, thank you. Another question from student is, could you please clarify about the stability issue of enzyme-based biosensors?
Stability of enzyme-based biosensors.
Yes.
Yeah. So first of all, since enzyme is a protein, right? And the problem with the enzyme is they get denatured very fastly, right? So you really need to figure it out whether, since enzyme needs a temperature which should be at 4 to 8 degrees centigrade in most of the cases. So in that case, you need to really figure it out whether enzyme is active or not. So before immobilizing on the electrode surface, you need to check the enzyme activity, perform the enzyme activity assay to determine the activity of an enzyme, and then you should go ahead with the immobilization on the electrode surface. First thing. Second thing, once you have already immobilized the electrode with the enzyme, having the single layer or maybe the double layer, then you need to be sure that it should be capped at the respective temperature, so that later on as well, the enzyme should not lose its activity. So simultaneously, you need to check whether, say, for example, if you have taken 5 days to check the enzyme activity, then again you need to verify after 1 month. Again, you need to verify after 2 months. And up to a certain time, say after 3 months, enzyme activity is most of the cases lost. So the drawback with the enzyme based biosensor is that you need to really maintain the temperature, maintaining the activity of the enzyme. Second thing, it's not related with the polymer-based biosensor. You don't feel any problem with the stability of the polymer-based biosensor, because when we perform the BET analysis for the molecularly imprinted polymer to check the size of the pores, we perform the gassing and degassing, and gassing can go up to 350 degrees centigrade for checking the surface area of the sieve. So now you can imagine that even if at the 300 degrees centigrade, our polymer retains its activity. So enzyme can be denatured, but the polymer can be. That's why we moved from the enzyme-based biosensors to the polymer-based biosensors, and we are getting really good results in that perspective.
Okay, thank you, sir. Next question is, can you please elaborate on the topic, specifically whether there is negative or positive charge generation on the microbes?
I'm sorry, I could not hear. Maybe I read it
Yeah,
one
sure.
by one, so that would. Please write here your question. Could you be okay?
From Anveshan.
Which one is it?
Anveshan at 433.
Okay, I am interested on bioelectric generation. Can you please elaborate on the topic, specifically whether there is a negative or positive charge generation on the microbes? So what we have been working on, that we have figured it out, since Uttarakhand is the region where you have a lot of biodiversity, and there are certain electrogens, electricity generating species which are not touched till now. So that's why we are creating library. So when the library work is finished, yes, of course, I can incorporate it. You can contact me later on. So the thing is, once we have developed this library, and once we have developed these genes, identified these genes, we will clone. We will develop a clone to generate a bioelectricity, and they will definitely give enormous current in a particular setup. So in Germany, there are a lot of research institutes as well as companies who have microbial fuel cells in a big sizes. So say, for example, this room, so 10 by 10 feet room can have a microbial fuel cells, and they have been working on constantly to generate a bioelectricity for the whole building. So now you can imagine how effective it is, that when we transform in three basic perspectives: selection of right microbial strain, second, working on the electrode surface, effective nanomaterials coating or effective electrode surface, and the environment where we are mixing in the microbial fuel cell. These three parameters we are focusing on, and of course you can contact me later on if you want to work on these ideas. So the next one is, so the next one is, as it is presented in the CV response of the gold modified biosensor, the final modified electrodes show the enhanced anodic peak current, whereas the cathodic peak current is not enhanced. Could you please explain this? So maybe I should, so which one is one? I guess it was in acetylcholine. Is it this one? I think. So I think, so yeah, it is actually in the potential state, you get the electrochemical response. And in some cases, the graph, what you see, it is the oxidation and reduction peaks. What you see cannot be always uniform. In some cases, it can be changed depending upon what can be your, what we say, the medium where you are evaluating the electrochemical signals. So in this case, I feel that why it was not getting that responsible signal which we were expecting, because maybe the oxidation peaks were enhanced through the electrochemical responses on the electrodes which were generated, and the oxidation peaks were much higher as compared to the reduction peaks. But it's kind of a, I would say it's also kind of a 3D imaging, where when you see, when you really closely look, then it should be oxidation and reduction peak in the equal quantity, but it's not always the case. Okay, so who is next? I think Yogesh has been done. I'm interested. Yeah, so I think nobody is left. Yeah.
I guess a question from Dipanshu Bhatt.
Okay,
Is
Dipanshu. I'm not able to see the Dipanshu's question.
it's
Acha,
yeah.
yeah, Dipanshu, yeah. Thanks for such an information. Because I want to ask that if a transducer is conductive, then why we should use ferroferry? It will it not interface with the materials redox peak? Rather than this, can we go for the PBS and other electrons carrier buffer? Yeah, this is very actually interesting question. So when we were developing microgens and electrogens, and previously we wanted to check that how much current was generated by the bacteria, we used ferroferry solutions because ferroferry is the solution where you get enhancement in the signal. Now the problem is actually you need a stimulus, a kind of a stimulus signal as part of the ferroferry where you get the signal which is higher. Now, when we evaluated that whether the electrons were generated on the electrode surface through the bacteria, then we moved to the broth culture and PBS to check whether if we are not using the ferroferry, then how much electrochemical response or electrical signals are being generated on the electrode by producing the electrons through the bacteria. And I can tell you that why we use ferroferry, because we really need a high threshold value. But you can, of course, go to the PBS, and I would recommend to go to PBS and other buffer solutions there, because again, ferroferry is an electroactive pollution, and it's not that much recommended when you're optimizing the things. So in my case, when I was working on the diabetic device, so there I won't use the ferroferry as a sample. I need to use the blood. So we thoroughly eliminated the ferroferry solution and then transform that priming. That priming you need to work on. That first you need to screen whether it's an electroactive component or electrochemical response you are getting, and then you should move in a real sample analysis. So in that case, you of course need to go with the PBS and other parameters, eliminating the ferroferry, right? Okay, I am interested in bioelectricity. Okay, that I have explained already. As it is presented in the biosensor peak current, this is also I have explained. Thank you for the wonderful talk, sir. I have a query which may not be that much relevant. Actually, sir, I am currently working on wastewater treatment using porous carbon-based nanomaterials. So could you please suggest that would it be best one if we collaborate the biosensing study in today's context? Yes, of course. So since I told you that we have been focusing on three different perspectives of microbial fuel cell. One is electrode material, one is microorganism, and third is the nutrient materials or wastewater materials, right? So in that case, I have published one review as well where we have changed a couple of nanomaterials in microbial fuel cells. So you can check that review. And porous carbon nanomaterial are widely used previously, but now people are thinking the substitute of porous carbon materials. Since porous carbon materials are having kind of a porous organization where you can get certain molecules entrapped in the porous region, and then you can have the higher electron generation. But I would definitely try some other nanomaterials, and of course we can collaborate. If you want to have another idea or you want to work with us, definitely I will connect with my PhD student who is working on this microbial fuel cell development. Okay.
I guess all questions are answered.
Swapna, I would have some questions to ask,
Yes,
so I need some time for that. Yeah, Dr. Utkarsh, this is a
Yeah,
really nice talk that you have presented. So I just have a couple of doubts. You have
sure.
mentioned that broadcasting is perhaps not the best of the ways to, do a coating. But if you see that the standard glucose monitoring setup
Right.
has a dropcasting of the blood on the paper strip, right? So do you really think that dropcasting
Yeah,
manipulation?
actually, it's a correlation. First of all, why I have told that dropcasting is not a really accurate approach, because when you see the enzyme-based biosensor, then when you immobilize enzyme on the electrode surface and when you load the sample on the enzyme loaded electrode surface, then the enzyme get diluted and it can be removed easily, because you drop cast and you dry it, right?
Yeah.
So drying cannot effectively let bind the enzyme to the electrode surface, and when you load the sample it can go. And that's the challenge what we also faced when in the beginning, when we were working on the drop casting approach for the glycated hemoglobin and glycated albumin. Now people in the glucose sensor, glucose oxidase, when it is used, so they actually are facing this problem. So what they had done in the glucose biosensor, that they coated the enzyme on the working electrode, and on the enzyme they have made a layer of mesh work so that the enzyme should not go out from the strip, and they actually entrapped the enzyme on the surface. So if you have as a laboratory model and drop casting the enzyme or other biorecognition element, then it will definitely go out. But if you really, after the drop casting, if you entrap that area with a mesh work or some kind of approach, then it will definitely work. But it will have certain challenges, that it will affect the real binding of the enzyme to the substrate. So you need to figure it out while optimizing the situation. So I would recommend to have a covalent linking of certain proteins and proteins as enzymes as well. When you have covalent linking, then the removal of enzyme is not that easy as compared to the drug casting.
Okay. And one another thing that I wanted to ask is like when you are working with these enzymes, whole cells and proteins and all the other more complex biomolecules, how do you exactly circumvent the impedance that are coming up? Is electrochemical impedance spectra is a more viable option than electrical resistance based?
Yeah. So I cross-checked that previously we were only working on the chronoamperometry, where we were working on the enzyme-based biosensors. But I can tell you that when we started working on molecularly imprinted polymer, the EIS gave really good data. So it is actually at par to the enzyme-based biosensor where electrons were generated. But when we have molecularly imprinted polymer, the entrapment of the template molecule or a specific molecule to the layer of polymer was really significant. And when entrapment were occur on the whole layer of polymer, and after the washing they were not removed, and even though we were washing for the 2 to 3 times, the effective binding, it was actually selective and specific binding that the entrapment could give the response in terms of EIS. So we then checked before nanomaterials coating and after nanomaterials coating. That's why we amplified the impedance response and getting the broader range of signal through the EIS study as compared to the resistance. We have never tried with the resistance and other approaches, but the mainly EIS impedance based approaches we have tried. So wherever we are checking the impedance which is caused on the electrode surface with couple of biorecognition element binding to the surface, the impedance EIS is really effective approach.
Okay. And regarding the aptamers, did you mention that there are extraterrestrial ones?
Yeah. So the Donald he researched for 40 years on the extraterrestrial aptamers. So he is really pioneer in this field, and he actually wanted to see whether if somebody is taking a drug in the space. So, for example, if somebody is having some problem and they are taking a particular drug, then they wanted to have a very rapid kind of device which they can bring to the extraterrestrial. And the reason why he recommended for aptamer, because a lot of research is going on that since the Muller experiment, when the origin of life came. So actually there were all the base pair sequences, and base pair sequences, they were combined in a sense that there are a lot of aptamers were discovered over the last 40 years. So he thought that aptamer based sensor could be a very effective device which the astronauts can bring to the space. And that's the reason we thought about the aptamers for the selection. And they have a very strong, one of the strongest binding affinity I could tell that you can get with the aptamers. And very specificity, very strong specificity. Because they are really say 50 to 60 to 200 base pairs, and they are arranged in a very specific arrangement so that they can bind with a particular molecule spontaneously with a high specificity. So that's why the reason why we selected the aptamers.
Okay, and just one last question. So do you really see the use of AI in the data?
I would say that I am not that familiar at this moment with the AI, and now I am working on it. I have collaborated with the really great AI people in my department, in my university as well. And it's sometimes, as a biologist, it sometimes goes from my head. And I am hopeful that when I recently started working on AI integration with our devices, so I'm trying to learn the things, and hopefully AI could definitely give the different perspective when we are working on specifically the designing part. So once we have a properly well documented AI approach, where we want to select which nanomaterials I would like to utilize, which biorecognition elements for a particular biomolecules I would like to utilize, that integration can be effectively given by the AI tool. And that is what we are working on, that app where we can have this possibility with the biosensor that which one you want to select for the design.
So it's basically the material design and
exactly. For that moment. And secondly, with the app development and when we are transforming the biosensing device to the mobile integration, there we are using the AI.
Okay. Thanks, Utkarsh. Thanks a lot for a wonderful talk.
Thank you for the invitation, Chaitanya. I am really thankful. I really appreciate that you are doing great job to guide a lot of students in this perspective. And hopefully, so by the way, we are also organizing one conference in 2026 in April. So it's a congress, big congress. And one of the special session we are doing in the electrochemical sensing, where a lot of electrochemical companies will be coming. So I will invite you for the talk as well. So
when
it's in April. It's in April. Sorry?
Okay. When is this happening? Next April?
Next year in April 2026.
Okay, that would be wonderful.
So a lot of people will be benefited with your experience as well.
Sure, thanks.
Okay, so have a nice weekend. All of you, thank you so much.
Thank you, sir, and
Bye
thank you
bye.
all the participants for joining us today. Thank you, ma'am. Thank you, Dr. Chaitanya.
Thank you,
Bye.
ma'am. Thanks for
Thank you, all of you.